Good afternoon, everyone. It's a pleasure for me to introduce Professor Christopher Parish. Professor Parish is currently a group leader in the Department of Immunology at Curtin School of Medical Research, Australian National University. He obtained his Bachelor degree of Science in 1966 and his PhD in 1969, both from the University of Melbourne. He completed 2 postdoctoral trainings, one from 1969 to 1971, here in the John Curtin School of Medical Research, and the second one from 1971 to 1973 in the Sir William Dunn School of Pathology, University of Oxford. During the last 35 years, he has investigated various aspects of the immune system. In the early 1970s, he was the first person to demonstrate that cell-mediated immunity and antibody formation are mutually antagonist responses, an observation that underpins the Th1-Th2 paradigm in immunology. He has also made a major contribution to the development of novel therapeutic anti-cancer agents through his investigation of the role of heparan sulfate, an anionic carbohydrate, in cell migration and invasion. These studies have led to the development of an anti-cancer drug that is now called mupirostat, which is currently undergoing phase 3 in clinical trials. Professor Parish is a consultant to a number of Australian biotechnological companies, and his expertise is frequently sought as a reviewer and assessor for journals and granting agencies, both locally and internationally. Professor Parish has an outstanding vision for developing commercial outcomes of his research, as evidenced by 25 successful patent applications over the last 20 years. 4 biotech companies have been built up with the assistance of Professor Parish and his research programs, and they are already providing great benefit to human health. He is a member of prestigious scientific societies. From 2003 to 2004, he was the president of the Australasian Society for Immunology, and he has also received several prestigious prizes, including the Goschall Medal that was awarded by the Australian Academy of Science in 1979, the Clive and Vera Ramanciotti Medal for Excellence in Biomedical Biosciences in 2005, and the Rowley Medal in 2007. The title of Professor Parish's presentation today is Emerging Cancer Therapies. Please join me in welcoming Professor Christopher Parish.
Thank you for the kind introduction. Well, this evening I'm going to divide my talk into 2 sections. The first part, which is indicated on the next slide, the first part is the main subject of this presentation, emerging cancer therapies. But I thought, since this public lecture is associated with a Translational Medicine conference, that I should also give a short summary of my experiences in translating my research findings into the clinic. It's quite a rocky road, and I think some of you may be interested in what's involved in that whole process. But first, let's concentrate on the major theme of this lecture, which is on emerging cancer therapies. Now, one question I'm frequently asked by friends, scientific colleagues, laypersons, is why is it that a cancer cure has been so elusive? Why is it that over 100 years or more there's been many very clever people working in the field of cancer, and yet our progress has been somewhat disappointing? There are some successes, there are some treatments for cancer that are very successful, but there are a lot of tumours that are still untreatable, essentially. Once the tumour metastasises to a distant organ, the prognosis is very poor. And so the question really is, why is it that it's been so difficult to generate cancer cures? Why is it so difficult that there isn't a penicillin for certain cancers, for example? And I think the major reason for this, and a lot of people agree with me on this, is that cancers are inherently genetically very unstable. They are able, particularly the highly metastatic, highly invasive cancers, their whole genome is in a state of huge flux, such that the population of cancer cells contains mutant variants that can almost evade any chemical, biological, immunological strategy that we throw at them. they're a moving target. So quite often some of the cytotoxic drugs that have a fairly bad name, bad reputation for cancer treatment because of their side effects, initially their effects in controlling the tumour are quite spectacular, and one gets regression of a cancer quite rapidly in many patients. But then gradually the cancer re-emerges, and when it re-emerges it becomes drug resistant. In fact, we end up having cancers that are multi-drug resistant in many patients. So how can we get around this problem, this inherent genetic instability, which means that we've got mutant cancer cells that are evading most of the therapies, particularly the monotherapies that we attempt to use to treat it? It also means that a monotherapy, a single treatment for cancer, is very unlikely. We'll need to have combination therapies in order to be able to control cancer, whatever approach we use. But firstly, let's look at ways in which we can overcome the genetic instability of cancer cells. What approaches can we use? Now, one obvious one, which seems very simple but isn't as simple as it seems, is just identify processes and block them that are essential for cancer cell survival and spread. Now, the problem with this approach is that often certain processes that are essential for cancer cell survival and spread are also essential for the functioning of the body, the normal functioning of the body, so that a drug that blocks a particular process may well be lethal for the patient as well. So we have to be able to identify unique features of cancer cells that separates them from the majority of healthy cells, which allows us to target them selectively. And also identify processes that are essential for the cancer cell survival, and I'll give you examples of that in a moment. The second approach is an indirect one, which is not to try and kill the cancer cells directly, because we know if we have a cytotoxic drug that is very good at killing a particular cancer cell by, say, interfering with DNA replication, we very rapidly select for variants that are not susceptible to that drug. But a lot of host components are required for solid tumours to grow. It's often referred to as the stroma surrounding a tumour, or the microenvironment surrounding a tumour, and that has to be of the appropriate type in order for the tumour to grow. So another approach is, rather than to target the cancer cells directly, to target the host components, the stromal components that are required for cancer survival and cancer growth. And the third approach is to harness the immune system against cancer. This is probably the most controversial. Many immunologists have thought that there possibly is an immune response to tumours, but it is of irrelevant nature. It's an epiphenomenon and doesn't play a role in cancer control. But there's increasing evidence arising now to suggest that the immune system does recognise many tumours as foreign, and that this immune response actually influences the growth of the tumour, such that by the time a tumour has been identified and by the oncologist, it has been immunoselected to an extreme degree, such that it evades the immune system of the patient. And I'll come back to that point a bit later in my talk, and how we can get around that problem and harness the immune system against cancer. So let's go through each of these approaches for overcoming this genetic instability of cancer and developing new cancer therapies. First is blocking processes essential for cancer cell survival and spread. Now, the next few slides just summarise various steps in the process of a tumour cell escaping from a primary tumour and metastasising to a distant organ. The primary tumour in most cases isn't lethal for the patient, except if it's in the brain or in some incredibly important organ. It's usually the metastases that cause the problem. If we could stop tumour metastases, in many cases the problem associated with cancer would be solved. And in fact, the most aggressive, most malignant tumours are the ones that metastasise. So trying to control tumour metastasis is a very important aspect of controlling cancer. Now, this diagram here is actually showing a primary tumour escaping into a vessel next door to the tumour, but often tumour cells also escape into the lymphatics, but they all end up in the bloodstream, where they can recirculate and get trapped in a distant vessel, as indicated here. And in this case, the tumour cells, and this happens in many cases, they form a microclot, a microthrombus around them, which favours their entrapment in small vessels and distant organs. Now, fortunately, this process is very inefficient, but the tumour cells then have to find their way through the blood vessel wall. And that process at the top diagram there, I'll see if I can use this. If this is working, yes. This top diagram here shows initial adhesion of the tumour cells to the endothelium. Endothelial cells are the cells that line the blood vessels. And then eventually the tumour gains the ability to degrade the basement membrane underneath. This blue layer indicated there, which is the basement membrane, which is a very major barrier for the passage of tumour cells through the blood vessel wall. But they need to deploy a range of degradative enzymes to break down this basement membrane, and then they eventually migrate into the tissue. Now, this process isn't unique to tumour cells. Leukocytes, white cells, use this approach to enter tissues when they're combating infections. And so it's really a process that has been hijacked by tumour cells, this metastatic process. In fact, it's one of the reasons I first became interested in tumour metastasis many years ago, because I realised that it very much resembled the same processes that leukocytes use when they migrate into tissues. Now, this basement membrane I've just highlighted there that underlies the endothelial cells lining blood vessels really does represent a major barrier for cells entering tissues. And in fact, it acts as a barrier for molecules entering tissues. It's a molecular sieve. Larger proteins in the circulation actually can't get into tissues due to this basement membrane. So as you can imagine, it's a major barrier to cell entry. The figure on the left is a scanning electron micrograph of a basement membrane. You can see that there are cells above it, and beneath it is the loose extracellular matrix, which is associated with tissues. But that basement membrane, which on the right-hand side is composed, and then I've got a sort of schematic there of what it's composed of, which is of a complex mixture, almost like a barbed wire entanglement, of collagen type 4 and laminin, which are proteins, and heparan sulfate proteoglycan, the blue molecule in the structure. Now, that blue molecule is made up of a polysaccharide called heparan sulfate and a protein core. It's the heparan sulfate, the polysaccharide chains, which play a crucial role in holding the whole basement membrane together. And if they're lost, the structure collapses and cell migration through the basement membrane is favoured. Now, an important breakthrough we had a number of years ago in my lab was we realised that an enzyme called heparanase is essential for cancer spread or metastasis. And the way this enzyme works is that it degrades heparan sulfate. And this is the way it does it. I think there was a pointer around here earlier that seems to have disappeared, actually. Claudia, do you know where the pointer is? Because there was one here when I came earlier. Sorry for the interruption. There was a laser pointer earlier, but it seems to have disappeared. Anyway, if you look at the structure here, the cleavage of heparan sulfate by heparanase. Heparan sulfate is indicated by that yellow line in the centre there, is a very linear polysaccharide made up of glucose molecules. And these glucose molecules are modified in particular ways so that where that jagged line is where they have sulfates and various other variations in structure occur. Now, heparanase is an enzyme that is able to cleave or break the heparan sulfate chain in very unique sites along the structure, such that large fragments of heparan sulfate are released from the basement membrane. But this is enough to, if you like, it's a bit like a barbed wire entanglement where you've got wire cutters where you just cut the wire in particular key points. And that allows the heparan sulfate to be released from the basement membrane and causes the basement membrane to become very much destabilised. Thanks, Claire. So this destabilisation that occurs, and here we have the heparan sulfate chain, as I mentioned, being cleaved by heparanase in these particular points, which results in fragments being released. Now, there's another feature of heparan sulfate which I haven't mentioned, and that is that there are a lot of growth factors that have an ability to bind to heparan sulfate and are actually sequestered in basement membranes waiting to be released. So when heparanase comes along and it's produced by a tumour cell or an invading leukocyte, it not only breaks down the heparan sulfate and aids cell invasion into the tissue, but it releases these growth factors that are sequestered in the matrix, the basement membrane, and makes them available for wound healing. Now, that's great if you want wound healing and combating an infection, But if it's a tumour cell using heparanase to pass through a blood vessel, that's bad news because it actually provides growth factors which aid the growth of the tumour and also aid the growth of other aspects of tumours, which I'll come to in a moment. Now, for those who are interested in the finer details of this enzyme, it's an endo-beta-glucuronidase, my scientific colleagues in the audience. Heparanase itself, when it's first produced by cells, it exists as this long-chain polypeptide that is enzymatically inactive, but then has to be activated by removal of this part of the molecule to form this heterodimer structure, which is the active enzyme. So heparanase can also be controlled by interfering with the enzymes that generate the active form of the enzyme, like this molecule called cathepsinL, which is another interesting aspect of heparanase. The structure of the molecule, its three-dimensional structure hasn't still been defined, but I thought I'd just show you what it looks like, we think it looks like. And it's this structure which has all these sort of helical structures around the outside and what's called these beta strands inside. And it's this hole in the middle, which is where the heparan sulfate enters and is cleaved by the enzyme. And this is another picture of the three-dimensional structure with these red molecules here. They're key amino acid residues involved in the active site of the enzyme. Now, when we discovered that heparanase was involved in tumour metastasis, and we then went about cloning the enzyme, it took 15 years to clone this enzyme. It's a horrendous enzyme. There were many other groups in the world, and there was actually an Israeli group at the same time as us finally cloned it and reported it in 1999, and we published back-to-back papers in Nature Medicine. It was one of the most important achievements of my career. But what we found with this enzyme was that there was only one heparanase gene in the mammalian genome. Now, one would have expected with an enzyme so important as this that's involved in breaking down heparan sulfate and aiding cell invasion, releasing growth factors, that there'll be redundancy in the system. There would be other enzymes that could do it, but there's only one enzyme that can actually act as a heparanase. There's no other enzyme that can do this. And we've looked for enzymatic activity as well as for genes that have similarity to this heparanase gene. And our group and other groups have come to the conclusion there's only one enzyme of this type. So this makes it an excellent drug target. This is the sort of thing we're after in the cancer area, a very important enzyme involved in cancer metastasis, and there seems to be only one. There doesn't seem to be any other enzyme that can do the same job. So we then went about developing a sugar-based, a heparan sulfate mimetic-based inhibitor of heparanase as an anti-cancer agent. Now, I should mention that the basement membranes I showed you earlier, they're made up of protein as well as a lot of protein components of the basement membrane. And there are enzymes that degrade that as well. But as far as a drug target, that's quite difficult because there are something like 30 different proteases that have been identified that can break down the proteins that make up a basement membrane. But there's only one heparanase. And the two enzymes seem to have to act together, the protease together with heparanase, in order to aid breakdown, solubilization of the basement membrane, and tumour cells being able to migrate through it. So we developed an inhibitor, Metastasis or PID8. Metastasis, a terrible name, but it turns out that Trojan Industries, as they were called at the time, our commercial partner, when they wanted to register this drug, it's an international committee that gives it the name. The actual inventors, the company, has no say in what it's called, which surprised me. So they came up with this real tongue twister, which is very easy to forget and difficult to pronounce, but Metastasis is what it's called. But PID8 is what it was originally called, PI standing for Trojan Industries, and 88. It wasn't the 88th compound we tested. Trojan has a lot of Chinese shareholders, and 8 is a very auspicious number in Chinese culture. So 2 8s meant this drug will have to work. So this is the structure of it. It's made up of mainly of a pentasaccharide and a tetrasaccharide of mannose, which is like glucose. Each of these is single glucose-like structure, and it's decorated with different sort of sulfate groups in different positions on the molecule. Its name is phosphomannopentaose sulfate and phosphomanotetraose sulfate, which I expect you all to remember when I ask you it after this lecture. Now, PID8 and heparanase inhibitors in general were found to be very potent inhibitors of tumour metastasis. This is an acute assay where we're looking breast cancer metastasis to the lung in a rat. Each one of these nodules here is a lung metastasis. So this is a very nasty metastatic breast cancer. But if we administer our heparanase inhibitor at the time when we know the tumour cells are being released into the circulation and spreading to the lung, we can essentially totally block tumour metastasis to the lung and to other organs as well by this heparanase inhibitor. The way we believe heparanase inhibitor works, and we've got quite a lot of evidence looking at tumour cells in animals that have got tumour metastasis occurring to try and see what effect PID8 has on tumour metastasis. And we can show that the tumour cells interact with the blood vessel wall as normal, but they can't get through this basement membrane. They just sit there. And the heparanase enzyme, which helps them get through, is inactivated. PID8 actually binds to the catalytic site of heparanase and is a non-cleavable substrate, so it just sits there and inactivates the enzyme, such that the tumour cells eventually, we believe, can't survive in this hostile environment of blood flow where there's a lot of shear forces, so they eventually get eliminated. So that's the first example I'd like to give you of the sort of approach, and I'm using examples from my own research, where you go for a target that's unique, or that's, if you like, the Achilles heel of tumour metastasis in this case, and the side effects of a heparanase inhibitor are fairly minimal. I'll come back to that probably at the end of my talk, why some of these drugs don't show side effects even though they seem to be attacking certain aspects of inflammation that you might think may not be so desirable. So the second area I'd like to talk about is attacking these host components that support tumour growth, the tumour stroma that is required for a solid tumour to grow. Now, one area that's caused a great deal of interest over the last 30 years and has been pioneered by Judah Folkman, a very, very clever surgeon at Harvard University. This is targeting blood vessels within tumours. And what we have here is a vascular cast of all the blood vessels that are present in fat tissue, adipose tissue. In this case, the vessels have been imaged by a plastic that sets within the vessels, and then the cells surrounding the vessels have been digested away, such you can see the vascular bed that a normal functioning tissue possesses. And you can see that there's an incredible network of blood vessels that are required for the normal functioning of this tissue. And this makes sense. It's a metabolically very active tissue, adipose tissue. It needs oxygen and nutrients, and it needs toxic byproducts of metabolism to be delivered and removed by the blood vessels within the tissue. Now, solid tumours are no different to normal functioning tissue. They need an active blood supply. They need a blood vessel network just like this. And once the solid tumour grows beyond 1 to 2 millimetres in diameter, it requires the induction of new blood vessel growth, or the tumour will not grow beyond that size. It does that by a process of angiogenesis. Angiogenesis is the sprouting of new blood vessels from established vessels. This is a very schematic diagram showing new vessels that are sprouting from an established vessel nearby a tumour that's developing. The trigger for this angiogenic process is hypoxia, lack of oxygen in a tissue, and it turns on a whole range of pro-angiogenic processes which tumour cells can produce just as well as damaged tissue can produce. So it's a normal physiological response, which again, tumour cells have hijacked for their own benefit. And this is a very important feature of cancer, is that cancers don't discover new biological processes. They just use normal biological processes in the wrong place. So, of course, there's a very attractive possibility is if one can develop inhibitors of angiogenesis, they would arrest tumour growth. A very important development for our own research at the John Curtin School was that we developed a very simple test tube test for angiogenesis. Now, screening for angiogenesis inhibitors is not easy. Most of the studies involved animal work, which was ethically quite tricky, using the cornea, the eye, for example. And so we looked for an in vitro assay, which was much easier to perform and ethically more acceptable. And in fact, we developed an assay which used human blood vessels from human placenta for this angiogenesis assay, which I'll show you in a moment. And we then use this to screen for inhibitors of angiogenesis. This assay has been incredibly useful for the last 15 years, where we've identified a lot of angiogenesis inhibitors, as well as an angiogenesis enhancers, which can be used to enhance wound healing. But in the talk today, I'm talking about inhibitors of this process. And heparan sulfate, small heparan sulfate structures, are also inhibitors of angiogenesis for reasons that I'll explain in a moment. So this is a picture of our angiogenesis assay. We have here a fragment of human placental vessel, which has been embedded in a fibrin gel, so in a normal physiological clot. And then over the next 10 to 14 days, one gets the outgrowth of mini vessels from the severed ends of this vessel fragment. This is after about 14 days culture here, but we can use other vessel fragments from animals as well and get just a similar result. But this is human placental vessel. So then we can screen for various inhibitors. This is just the control culture. And if we put in certain heparan sulfate mimetics, we find that they're very potent inhibitors of angiogenesis, as indicated here. Now, why does heparan sulfate come up again as being important? So far, I've just talked about it as being a barrier for cell migration through basement membranes and as a reservoir of heparan sulfate binding growth factors that are released by heparanase. But we also know that heparan sulfate plays a very important role in growth factor action. And the way it works here is this is a diagram of a growth factor called fibroblast growth factor, which binds to a fibroblast growth factor receptor on cells. And this receptor is on endothelial cells involved in blood vessel growth, but it also can be on tumour cells themselves. And the growth factor, when it binds to its receptor, it causes it to dimerize and give a signal to the cell to start proliferating. This interaction of the growth factor with its receptor, however, is enhanced when heparan sulfate is present. And the heparan sulfate cross-links the FGF with its FGF receptor. There's probably about a dozen growth factors at least that have been identified that require this heparan sulfate fragment to cross-link them with their receptor. And without that, you don't get such a stable growth factor receptor interaction, and the growth factor is nowhere near as effective at triggering cell proliferation. So what we did was to design tiny heparan sulfate fragments, not as big as this, that get in the road of either the FGF binding to heparan sulfate or the growth factor binding to heparan sulfate and stopping formation of this ternary complex which triggers cell growth. And the compound that we identified is this PID8. Now, this isn't an accident. The whole screening program was aimed at identifying a molecule, a heparan sulfate mimetic, as we call it, which could simultaneously block heparanase and also interfere with angiogenesis growth factors or growth factors, particularly fibroblast growth factor and vascular endothelial growth factor, which has been shown to be very important in angiogenesis. And it's this PID8 structure that has that double hit action. So the beauty about this sort of compound is that we're targeting cancer at multiple sites simultaneously with a single drug. We're not just inhibiting metastasis, we're not just inhibiting cell invasion, release of growth factors, but we're also inhibiting angiogenesis. So PID8 or mipafostat is a double hit anti-cancer drug. It inhibits blood vessel growth and therefore starves the cancer of a blood supply. And so it stops primary tumour growth, but it also inhibits cancer spread, or metastasis as it's called, by blocking the enzyme heparanase. And the nice thing about these molecules is they tend to have minimal side effects. Patients that have been treated with PID8 have no feeling of illness whatsoever. In fact, I've heard patients say they've never felt so good because I suspect they anticipate some reaction when they're given a new anti-cancer drug, but these drugs are very subtle in the way they act. So how has mipafostat or PID8 performed in cancer trials? Well, the phase 1 clinical trials were completed some years back in healthy volunteers and cancer patients. And the drug was well tolerated, and in fact, counting all the cancer patients that received this drug, there's something like 20,000 doses of PID8 now being given to cancer patients. Now, the most promising phase 2 clinical trials, remember phase 1 clinical trials are just establishing whether your drug is toxic or not, getting the maximum toleratable dose that a patient can have of a particular drug and working out the drug scheduling. If you have efficacy, that's a bonus in phase 1. Phase 2 is when you really test out the anti-cancer activity of your drug. The best results are obtained in melanoma, where it's been up to a 25 to 30% response rate. That means, by response, it means the tumours stop growing or even regress. Well, it's generally just stop growing. And the most spectacular result has been in post-resection hepatocellular carcinoma. I'll explain to you in a moment what that means, where there's been about 35 to 40% reduction in recurrence. As a result of these phase 2 trials, there's a phase 3 clinical trial that Progen is about to commence in hepatocellular carcinoma patients. And phase 3 is where you have a larger number of patients in the trial, just to get better statistical assessment of the efficacy of your drug and to satisfy all the regulatory agencies that your drug really is worth registering. Here's a bit of patient data. These are CT scans of the melanoma patients fairly early on in our PIDA trials. There are 2 patients. The top patient is a patient where before treatment they were treated for PIDA for 3 months. This is the tumour in the lung. And after 3 months, these patients, I should add, are stage 4 melanoma. Stage 4 melanoma, the prognosis is very bad. Tumours are growing quite rapidly. So to get stable disease is quite an achievement, because the life expectancy of these patients is about 6 months. So in this patient, after 3 months, this is the images after 3 months here, there was actually, they call it stable disease, but actually I think the tumours are getting smaller. And I realised some years later that stable disease often covers up to 50% tumour regression, or 30% tumour regression before you say patient response, which means regression. So there's quite a big error bar there in terms of what is regarded as regression. But this patient continued treatment, this is 17 months later, and in fact both these patients I think have continued treatment with P88 to the present day. Certainly one of them I know has for sure, and that's 9 years they've been treated with this drug, and when they were first diagnosed they had 6 months to live with their particular melanoma. This is another patient here, the one that's I think received it for 9 years, and they had after 27 months of treatment, P88 started to show, initially it was stable disease and then regression began to occur. Now this is typical of what one sees with anti-angiogenic drugs. They're not going to cause regression of the tumour that's already present. You can't deconstruct the vasculature or the blood supply that's already present, but you can prevent new blood vessels growing into the tumour, and that's what these anti-angiogenic drugs do. And so that's why stable disease is what you expect initially. But it has been noticed that after treating patients for long periods with these drugs, that regression of the tumour begins to occur. And it may well be it's because the maintenance of the already established blood vessels within the tumour is also being interfered with by prolonged treatment. These are the data that we obtained in the hepatocellular carcinoma phase 2 clinical trial. Now, in hepatocellular carcinoma, primary liver cancer, the main treatment is surgery. The primary tumour is removed from the liver, and then there's really not much treatment given. The prognosis is very poor. 50% of patients get recurrent disease within 1 year, and those patients, most of them die within the next 2 to 3 years. So it is a very good cancer to see whether P88 can change the course of disease, because the patients have had their primary tumour removed. They don't have much tumour mass left. And so P88, which stops tumour growth and metastasis, should work well in these patients. So in this trial, patients had their primary tumour removed, and then 5 weeks after surgery, P88 treatment was commenced, and then recurrence of disease was followed. So this is showing the percent of patients that are disease-free at different stages after treatment. You can see in the control group by 50 weeks, the P88 treatment was for 30 weeks, incidentally. By the end of the year, after surgery, as in historical statistical analysis, about 50% of the patients have got recurrent disease, whereas those that receive P88, a much lower percentage of patients have got recurrent disease. And you can present the data slightly differently. You can look at how long it takes for the 70 percentile, or for 30% of the patients in the control group, untreated group, to have recurrent disease versus the P88 treated, and there's a very big delay. So those patients who actually did develop disease develop it at a lot slower rate as well. Plus, there's a whole lot of patients who haven't developed disease at all at this stage. So this is a very encouraging result. And as I said, phase 3 clinical trial is about to commence with this drug. So the final area I'd like to talk about is harnessing the immune system against cancer. Can we actually do that? As I mentioned earlier in my talk, there's now emerging evidence that there is an immune response against cancer, and we know that makes a lot of immunological sense now because cancer cells have mutated proteins within their genome. They scramble their genome, they produce many aberrant proteins that are different to what would have ever been seen by the patient's immune system, as well as upregulating proteins in very strange places that are not normally expressed in tissues, very rarely expressed. And so it's now thought, and there's quite a lot of data to support this concept of cancer immunoediting, such that early on, after a tumour arises, the immune system can respond to it. And in fact, a lot of the cases, the tumour may even be eliminated by a very active immune response, which involves all arms of the immune response, both innate and adaptive immunity. However, some cancers, due to their particular immunological makeup and their immune evasion ability, the number of mutant cells that can arise that can evade the immune attack, reach an equilibrium state where the rate of tumour cell destruction is comparable to the rate of tumour growth. But then we get escape, where the mutant cells that have evaded the immune response take over and the tumour becomes very evident. And it's at this stage where the tumour is presented to the oncologist. So you can see that by the time a patient realises that they have a cancer, it has already evolved to evade the patient's own immune system. So the challenge is to reinvigorate this immune response against the tumour, which could well have controlled it for many years. In fact, there's some evidence that this sort of tumour dormancy that can occur in this equilibrium state can exist for many years and has an immunological basis in many situations. So one of the novel developments in my laboratory, I'm an immunologist primarily. I got into cancer metastasis and angiogenesis by following my nose on trying to understand how leukocytes get into inflammatory sites, but immunology is my number one passion, and so we were very keen on identifying immune responses that would be very difficult for cancers to evade. And we found that there was a particular type of an anti-cancer immune response which was difficult to evade, that involved, well, I'll show you in a moment, but involved an explosion of particular leukocytes which killed bystander cancer cells. So it would be very difficult for the cancer cells to evade this type of immunity. And we've also now developed a vaccine that can use this potent anti-cancer immune response as well as other well-known anti-tumour responses. So this gives you an idea of the sort of results we can get. This is a pretty gross slide, but this is melanoma metastasizing to the lung of a mouse. It's black because the tumour cells are containing the melanin, the pigment melanin. That's why they're all black like this. It's got hundreds of metastases, this animal. If we take animals that are not quite as bad as this, but almost as bad as that, and develop this immunotherapy where we're transferring, in this case, T cells that have the right type of immunity against the tumour, we can totally clear the tumour dramatically. There's a few tumour cells, a very small number of tumours left, but it's an absolutely dramatic clearance of the tumour. And we found that this immunotherapy that we've developed is a bit different to ones that other people have been involved with. It involves a response against tumours similar to chronic asthma. Chronic asthma not only just involves mast cells with IgE on them binding allergens and producing all sorts of molecules that cause the allergic response, but chronic asthma has the problem that it involves recruitment of eosinophils, a sort of less poorly understood white cell that is thought to normally combat parasitic infections. It gets recruited into the lung, degranulates and causes lung damage. What we've managed to do is to hijack this type of response inside a tumour and get these eosinophils to degranulate inside the tumour and kill the tumour cells in the tumour site. So they explode and kill many neighbouring cancer cells within the same environment. And this effect can be within a matter of 72 hours. You can get total clearance of the tumour within 3 days. It's very dramatic. The problem with it is it involves taking T cells out of the animal, educating them appropriately in vitro and putting them back in. And that's not really a long-term strategy. So we've managed to actually develop a vaccine strategy which turns on that type of immunity as well as other forms of immunity. And this vaccine we've developed called Lipovaccin, and it's a vaccine that targets cells called dendritic cells, which are absolutely essential cells for induction of immunity. And this is a vaccine that I've developed in collaboration with Joe Alton, an ANU academic, a different part of ANU to the John Curtin School. Now, dendritic cells are master regulators of immunity. They represent a very small population of white cells within lymphoid organs and in tissues. In the lymph node, for example, and spleen, they probably represent less than 1% of white cells present in that tissue. But they're very uniquely placed. They're positioned within the tissues in a position where they encounter pathogens very effectively. And they also have on their surface an array of receptors that have evolved over millions of years that are specific for unique molecular patterns associated with many of the common pathogens like bacteria, viruses, fungi, and parasites. Huge range of these, and they're still being discovered, hundreds of them, of these receptors called pattern recognition receptors, which recognise these unique structures that are associated with pathogens and not with our own tissues. So what happens then with these dendritic cells is when they encounter a pathogen, they take it up, they process it and present it to the immune system, to T cells, and the very receptors that are involved in recognising pathogens activate these dendritic cells, tell them, hey, we're interacting with something foreign, it looks dangerous, respond to it. Interestingly, these dendritic cells are also continually taking up self-proteins, self-cells, and presenting them to the immune system, but they maintain our immune system such that we don't develop autoimmunity, so our immune system doesn't respond to self. Unfortunately, what happens in cancer is that tumour cells take advantage of this balance between dendritic cells turning off immunity and turning it on, and often develop an environment where the dendritic cells are actually turning off immunity to the cancer cells. And this can occur in the microenvironment of the tumour and even in the lymphoid organs of a cancer patient. So what we decided to design was a vaccine that targeted dendritic cells, and it's based on what's called liposomes, which are these sort of membranous sacs that are depicted here. They're usually uni-layered, like indicated here, but they can be multi-layered, and they're very small, about 100 nanometres in diameter. They've been used widely for about 30 or 40 years to encapsulate various membrane-impermeant molecules, such as proteins, drugs. Doxorubicin, the anti-cancer drug, is delivered in liposomes to patients to limit its toxicity in tissues, and the nucleic acids have been incorporated in liposomes to introduce them into cells in processes called transfection. And the important feature of these liposomes is they can fuse with cell membranes and release their contents into cells. So they can act as really good delivery vehicles to any cell you like in the body. That's in theory. But normally, if you just to make a liposome and inject it into an animal or a patient, it is rapidly cleared because these phospholipids that we use to make up the liposome, these head groups here are recognised by scavenger receptors on phagocytes, engulfing cells in the body, and they're cleared within minutes. The reason for this is that these scavenger receptors are used for cleaning up dying dead cells, and so these liposomes are regarded as like dead cells and cleared by this phagocytic process. But what we've managed to do is to develop a very specific molecular glue which allows you to engraft onto the surface of liposomes any protein you like that can target the liposome to any cell you like in the body, in theory. And it's this structure here, I won't dwell on it, except to say this tail here inserts into the surface of the liposome, and this part here waves out in the breeze. This is the name of the molecule, which again I expect you to memorise, of course. And it's a triacetic acid-based structure which binds metal ions and then is able to chelate to particular amino acid sequences on proteins, what are called polyhistidine sequences, which are very rare, don't normally occur in nature at all, but we can genetically engineer onto the ends of proteins such that they have this histidine tag, which has a very high affinity for this NTA group, which is poking out from the surface of the liposome. So using this approach, we can take a liposome, incorporate into it small amounts of this NTA lipid, which binds the histidine tag proteins, and then engraft by their histidine tails proteins on the surface of the liposome. And so you can genetically engineer any protein you like by putting a histone tag on it and then engraft it on the surface of the liposome. What we've done is mainly use antibody fragments because that gives us enormous versatility in what we can target. But you could engraft other molecules. And these antibody fragments, we've concentrated on ones that target dendritic cells. You can also use this liposome to encapsulate antigens, foreign tumour antigens, and cytokines, and modify the dendritic cell when it is unloaded into the dendritic cell, so it activates it. These are called cytokines. So there's enormous versatility with this particular structure. Our cancer vaccine, however, is even more complicated, which involves taking our liposomes that have this 3-NTA lipid and fusing it with membrane vesicles prepared from, in this case, a melanoma cell line, but it could be from any tumour type, and making a hybrid liposome which carries hundreds. We've estimated there's something like 700 to 1,000 different proteins derived from the tumour present in this particular membrane preparation, which is fused to form this lipovaccin liposome, which has gamma interferon added to it to activate the dendritic cells. It's a very complicated structure, but it works extremely effectively, and we've done a lot of animal studies to show that this, I won't have time to show you, but this vaccine construct in animals can induce potent anti-tumour immunity that can cure animals of established cancer. Very dramatic effects. So we've managed to make a human version of this that targets a molecule called DC-SINE on human dendritic cells. And this is just a picture. Incidentally, these liposomes are nothing like as large as this. They're much, much tinier than that, targeting a dendritic cell, binding to DC-SINE, being internalised, and the dendritic cell being activated, and presenting the tumour molecules, foreign or the tumour antigens, to the host's immune system. So this slide here is just giving you a little bit of data showing how this targeting works. This is showing the peripheral blood mononuclear cells. These are just all the white cells out of the peripheral blood. And they don't have DC-SINE on their surface, whereas you can take certain dendritic cells that have lots of DC-SINE on their surface. This is a plot of density of a particular molecule on a cell's surface based on fluorescence. Now, if we take our lipovaccine vaccine, it doesn't bind to the normal white cells in the blood at all, but it binds brilliantly to these dendritic cells that have DC-SINE. This is about a 300 to 1,000-fold targeting. This is a log scale along this axis here. So there's a massive targeting of the liposomes to the dendritic cells by this particular DC-SINE targeting device. And this particular structure activates the dendritic cells if the vaccine has gamma interferon incorporated in it. So a phase 1 clinical trial has commenced with stage 4 melanoma patients in Adelaide in December last year with this particular structure, this vaccine, and we already have found the vaccine is very well tolerated as we expected, and not only that, but some efficacy has already been observed in the patients. Well, it's a bit early to comment on that too emphatically at the moment, but it's very, very promising. And of course, this vaccine can be modified to induce immunity against many different tumour cancer types, and in fact can be used to make vaccines against almost anything, actually. So it's a very interesting platform technology. So getting back to this whole idea of overcoming genetic instability of cancer, we need to block processes essential for cancer survival. Heparanase is a good example of that. Angiogenesis, inhibitors of that, is attacking components that support tumour growth. And lipovaccin is an example of an immune attack against cancer, which is very broad spectrum. We're inducing immunity of several forms, including that highly potent one I mentioned, that causes the eosinophils to be recruited into tumours. And we believe that individually or even a combination of these treatments, PID8 and the vaccine would be compatible, is a way of having a multi-pronged attack on cancer and much more likely for it to be successful. Now finally, in the last few minutes, and I'll skip through this quickly, but I thought I should share with you my experiences in translating my research findings into the clinic, firstly with PID8 and then with lipovaccin. Two very, very different experiences, I can tell you. Now, PID8 development, the first point I want to make is that these discoveries take a long time. I got involved in studying the role of carbohydrates in cell behaviour in 1975, which started my interest in carbohydrates and their role in tumour metastasis. had my first evidence in 1984 that sulfated polysaccharides, which looked like heparan sulfate, inhibit cancer spread. By 1986, heparanase had been identified as a drug target. And although we had no good inhibitors at that stage and the enzyme hadn't even been cloned, we didn't know how many enzymes they were, we knew very little about the actual target itself. So the first patent was filed in 1987, which covered heparanase inhibitors as anti-metastatic and anti-inflammatory drugs. And we then started searching for a commercial partner. The fledgling Anionic helped out at this stage. And we got $1 million deal quite quickly. 1986-87, for those in the audience who are a bit older, may remember that was a time when the stock market was going crazy. And we managed to get a very good deal. There was $1 million deal signed by a company that previously had been making diving bells, of all things, but was interested in getting into biotechnology. So it was planned to sign this $1 million deal on Monday, 19th of October 1987. You might say, why do I mention that date? Well, that happened to be the exact day that the stock market crashed. And the people never showed up. And we never saw them. The signing was supposed to be in Sydney, and they never turned up. We still don't know what happened to them, actually, whether they committed suicide or something, I'm not sure. But that was called Black Monday. The problem was that that put biotechnology back 5 years. It took 5 years before anyone was interested in investing in biotechnology in Australia. Recovered a bit faster overseas, but Australia was hit really badly. At the same time, just before this stock market crash, I had my first publicity about the research. It was decided by ANU that Anionic had been talking about our work, and they said, well, that wouldn't be a bad idea having a bit of publicity in the ANU Reporter. And the ANU Reporter said, well, yeah, that sounds great. And they put this article in the ANU Reporter, look a bit younger in those days. Anyway, that was the article. They said, gee, this story's pretty damn good. I think we should have a press release. And so they put together the press release, and then they said, well, the director of our school said, well, I think we should have a press conference. This looks like really good stuff. So we had the press conference, and it was on a Thursday. It was September the previous day, September the 24th. Here I am at the press conference. And it so happened that it was a very quiet day in Parliament House, and the whole of the parliamentary press gallery came to the John Curtin Scholar with 30 journalists, radio, TV, you name it. It was all there. Newspapers, they're all there. And so this story, which I expected to have one or two journalists strolling in to just have a bit of a chat, ended up circling the globe about 3 times in the next 48 hours, so that people in the US, my collaborators in the US said, hey, I think we heard a bit about your science this morning on the news. And so it just caused a media blitz. No idea. Just went everywhere. So we got lots of headlines. This was the one in the Age, quite conservative and quite acceptable. Canberra Times got a little bit ahead of itself. We didn't really have a cancer therapy at that time, but it looked pretty common. Sydney Morning Herald got the carbohydrate side of it quite right, but the Australian really got slightly just confused because we had mentioned that at that stage heparan and heparan sulfate could be purified from offal of animals, and that was our starting point, so that was where the drug was going to come from. So this media blitz was pretty scary because the phone was ringing non-stop, 100 phone calls a day, and we had to actually go down the coast to escape the media. But it turned quite nasty, actually, because at that time, commercialisation wasn't regarded as particularly kosher by a lot of academics. And I got attacked by some of my scientific colleagues. So a few days later, this article appeared in The Age where they started saying, well, I was overselling my work. It was exaggerated claims about this anti-cancer drug. It was really quite unacceptable. And the disappointing aspect of this whole article was that the actual press release and press conference was held before the work had been published. It was just about to be published. The patent had just been filed. So no one had actually ever seen the data. And in those days, there were no emails. You only relied on the posted article, which didn't come out for another 3 weeks. So these people were commenting on science they had not even seen. Nevertheless, it was pretty upsetting, and Anne Jackson-Nakano, the journalist who had originally written the ANU Reporter story, was very upset about what had happened, and so she decided to write a major piece for the Sydney Morning Herald putting my case that really I shouldn't be chastised for revealing to the world how exciting science. The public should know about what scientists are doing. It's silly to be hiding this matter. And so this came out and caused a bit of a stir with the scientific critics. So just over a week later, another article appeared in the Canberra Times from Feed Morris, who is my major antagonist and attacking me the most. And unfortunately, and I won't go into details, but at this stage, this article contained defamatory comments about me and the scientists in my group. This couple of paragraphs here, which was deemed to be defamatory, so we ended up suing the Canberra Times. So here I started my translation. My first experience for translating my research was that the company we had identified disappeared without trace. The first publicity I had about my research resulted in me being chastised by my colleagues and then ended up in a defamation action against the Canberra Times. So it wasn't a very encouraging start to translation. But things improved after that, and improved in a very strange way, very unexpected way. The New South Wales Country Women's Association came to the rescue. Now, and this is an article that appeared in the Canberra Times. They had seen all this publicity about me, and I think they thought I was the classic Aussie battler who was being chastised by everyone. And they said, look, we're really interested in your science. We'd like to raise funds for it. We have a medical research project we raise funds for every year. We would like to do that for your project. And I thought, oh, yeah, that's very nice. Thank you very much. And so they started raising money, and I thought, well, it'll be about a couple of $1,000s. They ended up raising almost $100,000 in a very short time. This was the article about the first cheque of $17,000. They came back and back and back, and that money allowed us to continue the work. Now, I should add, why was money so difficult then? Those were the days when the John Curtin School was block funded. We had no extra money. We were not allowed to apply the NHMRC or ARC for extra money. So we really had great difficulty if we had a new area, because I had a lot of other research going on at the same time. We had a new area of research we wanted to pursue and commercialise, and we just didn't have the funds available. So CWA funding allowed us to search for beta-heparanase inhibitors. And then the Commonwealth Government introduced syndicated research and development funding, which really, I think, and when one looks back on it, was a way in which banks could minimise their tax payments, really. It was a way in which they got a tax rebate if they invested in R&D. And unfortunately, I think it was somewhat rorted, but in our case, we really did do what was wanted. They provided us with quite a lot of money, which allowed us over the next 3 years to develop angiogenesis and heparanase assays, which allowed us to screen for the best compounds. And that attracted Progen funding. Progen got very interested in our work. It had reached a very interesting stage in 1993. And they funded our work for 8 years. By 1996, PID8 was identified and patented. Phase 1 trial in 98, cloning of heparanase reported in Nature Medicine in 99. The reason I mention that is this highlights the fact that sometimes my commercial colleagues used to say, well, if you're funded by commercial funds, source of funds, it's really not going to develop anything useful in terms of scientific publications. So that was exactly not the case. We published in the top journals throughout this period. Phase 1 clinical trials began in 2003. In 2007, we got that really exciting hepatocellular carcinoma Phase 2 trial result. So I thought everything was heading towards registration of a new anti-cancer drug, and everyone was very excited. Progen was planning a big trial in 60 hospitals in 13 countries of PID8 in hepatocellular carcinoma, and then disaster struck. The Progen board decided, in their great wisdom, that PID8 was no longer commercially viable. They weren't going to make enough money out of it based on their assessment of the market. Now, I won't go into the details of that except to say that liver cancer tends to be a disease of the developing world rather than the developed world. So, of course, a lot of the patients wouldn't be willing or able to pay much money for the treatment. So this was just devastating. Progen didn't even have the good manners to inform me that they were going to announce this. First I heard about it was from someone down the corridor who came along with the press release. So that was very unimpressive. But fortunately, Progen's had a lot of infighting amongst the board members and shareholders, and so finally a new board has been appointed, and now they've reinstated PID8 and plan to finish off the Phase 3 clinical trial. But the unfortunate part is that 2 years have been lost of patent coverage. We're closer to when the PID8 patent runs out. We've only got about 5 to 6 years to get this drug registered. If you get it registered in time, you can then actually request for a patent extension of 5 years, but it's getting pretty close to the line. There's still a distinct possibility that the drug may never actually get into the clinic, which would be dreadful. The Lipavaxin story is a much happier one. This started back in the 1990s. Again, it's an example where technology, the basis of the technology that's being developed involves many years of work. And then by 2002, Lipotec was founded. We had this engrafting procedure for targeting liposomes, but we hadn't really fully developed the best way of using the technology, but Lipotec was founded. It's an ANU-based startup company. It's in the Innovations building just across here. If it was daylight and those windows were open, you could actually see the building from here. So it's a local biotech company. The animal studies that we had carried out showing the efficacy of our dendritic cell targeting vaccine were published in 2004, but then we had the problem of re-engineering the whole vaccine construct such that it could be used for human use, because the vaccine we developed for mice couldn't be used in humans. We had to re-engineer it, and this involved collaboration with a company called Demantis in the UK, who developed domain antibodies which we used to target the dendritic cells and produced a domain antibody to DC-SINE. And this has been that one I showed you earlier, which is a very potent targeting molecule. This also resulted in a joint development arrangement between Demantis and Lipavaxin MM, between Demantis and Lipotec, it should be mentioned here, on the Lipavaxin MM project, so that the cost of development was being shared. Lipotec didn't have to share it to cover all the costs, and this is because, of course, Demantis was contributing the domain antibody IP and Lipotec contributing the liposome targeting IP. So over the next 60 years or so, the Lipotec crew particularly, and with the help of Demantis, developed this vaccine construct, and I must say that the Lipotec team has done a fantastic job. This has got 6 components, this vaccine, and I think it is probably the most complex vaccine that has ever been produced and injected into humans. And yet they've done it over in this building here at the Innovations building, and with, of course, other parts around Australia, producing the components of the vaccine. And the toxicology was also had to be carried out in primates to make sure that the vaccine is safe, and that all went according to plan. So now we have the... oh yes, I should mention before that in 2007, Demantis was merged with GSK, GlaxoSmithKline, a huge pharmaceutical company. We were very worried about this. We had very good relations with Demantis, but it's probably... Sorry? Can't hear me? Oh, that's unfortunate. I thought... So anyway, Demantis merged with GSK. I'll just make this thing a bit close. Demantis merged with GlaxoSmithKline, a major pharmaceutical company. And we were worried about this because we thought we would be dealing with big pharma and we'd lose contact with the Demantis staff, but it's actually been the best thing that's happened because we now have a joint development project with GSK as well as Demantis, and they have the wherewithal to follow the trial all the way through to phase 3 clinical trial and registration. And so the phase 1 clinical trial started in 2009, as I've already mentioned. So Lipotec also has a platform technology of other vaccines it's developing using this technology. So what lessons have I learned from my translational experience? Well, the first thing is choose your commercial partner very carefully. I mean, Progen was good at the start, but then I think it fell apart, and I'll tell you why I think it did in a moment. Second thing is, does the company have the resources to develop your intellectual property? Now, you might have thought Progen was a better bet than Lipotec, but Lipotec has actually probably developed the IP faster than Progen, even though Progen did support us well in the early years. So it's very much dependent upon the company, not necessarily how much money they happen to have. There's also now emerging non-commercial public good pathways for development. There's a few around, and they should be investigated so that you don't get into the problem I had with Progen, where it suddenly decided on commercial grounds, not scientific grounds, that your therapy is not worth continuing with. Check the R&D contract with a fine-tooth comb. You might say that's pretty obvious, but when the agreement with Progen was developed with ANU, the scientists were not allowed to look at the agreement because it had the banks involved, and they didn't want us to see some of their commercially sensitive information. But as a result, the R&D contract we had with Progen doesn't have an escape clause. So when Progen stopped developing PID8, we couldn't take it back off them in 2008. And finally, lastly, but most importantly, the inventors should be directly involved in the translation of their discovery. That's what's happened with Lipotec. Progen, we were very much involved at the early stages, but then they decided they didn't want us anymore when they had the drug. In fact, ANU wasn't even mentioned on their website as being involved in developing the drug. I think that was a major mistake because they didn't get our expert opinions all the way through the drug development stage. And that's why I think they faltered in 2008. So finally, all the collaborators involved in the work, huge number of people involved. I won't go through them all except to say that fantastic team at Lipotec, and a great team in my lab that have been involved in developing these therapies over the years. So I'll finish there and I'm quite willing to answer any questions. Thank you very much.
Thank you, Dr. Parish, for such a lovely and fascinating talk. The floor is open for questions.
Yes, Gavin.
Chris, why is a simple virus treated with a triple therapy, whereas cancer seems to be treated with sequential monotherapy? Why do you think that mindset is?
I don't know, really. I mean, HIV is really like cancer in my mind. It's a moving target. And so you need multiple therapies to get at it. I think it's probably we've been spoilt with viruses because some of the viruses are very hardwired in the way their life cycle behaves. But all the easy vaccines have been made. The difficult ones involve, in many cases, pathogens that have tremendous genetic variability during their life cycle, or even there's a whole lot of variance within the population of viruses that you can take, like influenza, for example. Yeah, I mean, it's a funny mindset, I agree.
Are you still blocked from applying for funds?
No, we're not. 2002, deblocking occurred, and my lab's never been so well funded.
Other anti-angiogenesis drugs seem to be having some bad press recently. I think Assay was mentioned. But your discovery works in an entirely different way, if I understand.
Yeah, I mean, you've got to be a bit careful with anti-angiogenics. I think one of the secrets of our drug development has been we haven't produced an inhibitor that's unbelievably potent. Some of the ones that are causing side effects shut down every single molecule, say an anti-VEGF antibody, for example. And so some of those molecules are required for normal vascular behaviour function, whereas P88 is a drug that probably knocks out 80% of that growth factor, 70% of that one. Now, tumour cells are pushing the boundaries. They're short of blood supply all the time, growth factor starved. They need every single molecule. So they're very vulnerable. Wound healing is a very robust process. It's a lot of extra capacity. You can knock out probably 70% of the growth factors involved in wound healing and still get normal wound healing. So I think that's probably the secret of our compound working so well, is that it hits a lot of things fairly well, but not too well. Yeah, I think the problem is, as been seen in a lot of situations in certain mouse models, you get fantastic data, but it doesn't translate into humans. But on the other side, there are situations where you get results in humans, and we're finding, and I can't comment on them because they're subject to some patent aspects, but we're finding effects with our cancer vaccine in humans that we sort of didn't expect, but are probably working in a totally different way to the way it's working in the mouse models we used. I can't comment on it, unfortunately, at the moment because we're about to patent it. But until we did the human trials, we never would have seen that. I think the other problem is that most of the cancers that are treated in patients have been growing in the patient for years. Most animal models you transplant a tumour that grows very aggressively within a few weeks. So you don't get the same microenvironment in these acute tumour models as you do in mice, as you do in a tumour that's developed over many, many years, have been immuno-edited, selected in a cancer patient.
Do you think that in the future you will be combining two strategies to...
Yeah, I do. I mean, I think going with one strategy in cancer is not the way to go. I think anti-angiogenics have actually been found to normalise the vasculature in tumours so that leukocytes can more efficiently migrate in and immune attack is more efficient. So I think PID8 could be combined with a vaccine, and they wouldn't be antagonistic, they'd probably be synergistic if not additive.
Your research, quite obviously, is focused on particularly around the immune system after cancer. Have you looked at implications
Before, sorry?
the implications pre-cancer? Obviously,
Yeah.
looking at prevention.
Yeah, sure. Yeah, you mean pre-treatment of PID8? Yeah. Yes, there are actually. There's a study over in the US where they actually developed a cancer vaccine to treat cancer patients, but they realised that it could be a vaccine that could be given to people randomly, just across the board to reduce their chances of getting cancer. That has been discussed and that hasn't been tried yet. It's a very difficult trial to do, of course. I mean, it would take many years to do it, and most commercial entities are not interested in it for that reason, which is a big problem in drug and vaccine development, is that there are certain long lead times that are not compatible with a commercial pathway of development. But anyway, getting back to your question, yeah, that has been considered, that there could be vaccination approaches that would reduce your chances of getting cancer in the future.
Does the PI88 affect the angiogenesis of normal healthy cells?
Yeah, well, that was I sort of answered that in the earlier question. Yeah, we expected PID8 would inhibit wound healing. We were quite concerned, and a lot of angiogenesis inhibitors, people have been concerned about that. We found that it doesn't affect wound healing at all, which was a surprise. But then we realised that to block wound healing, you need to really knock down growth factor action by a very high percentage, 95%. There are a lot of processes in the wound healing process that are very, very robust, much excess to capacity, such that you can give PID8 and it doesn't have any effect on wound healing but can inhibit your tumour growing and spreading, which is very fortuitous.
And have you seen any resistance to the drug?
Not yet, but it's early days. I mean, we have had people for 9 years on the drug, but certainly it's too early, I suppose, to say. That hepatocellular carcinoma trial, I don't know what the follow-up of those patients is, the ones that perhaps were resistant. Do they then suddenly get disease? And then we'd have to give them PID8 again and show that they didn't respond. So we just haven't got enough clinical data to answer that question. I would say it's less likely than most drugs because it hits so many targets simultaneously.
Any other questions?
So I guess this is very molecular, but the heparanase, because you show the 3D structure, does it have any regulatory interactions with the protein?
Does it have any what interaction?
Sorry, they have heparanase
Yeah.
in the mutant protein. You said that it encodes for a heparanase, which breaks the basement membrane. Is that what you said? The heparanase?
Yeah. Sorry, I don't quite understand your question, but I mean,
Show us a 3D
yeah, I'll show you the 3D structure. That's all theoretical structures at the moment. We haven't got the 3D crystals, but we've got a lot of molecular modelling that fits mutational data and all this sort of stuff. But the crystallising heparanase has been a major headache where no one's managed to do it yet. A lot of people have tried.
I was just wondering if it has any regulatory interactions with it.
Yeah, it's not fully worked out. There are some basic residues around the combining site where we think heparan sulfate binds, but that's pure speculation at the moment.
Thanks, Chris, for a great talk, and thanks for the comments about your experience over those years. Just a quick question. In the phase 3 trial that's being proposed, is it disease-free survival will be the primary efficacy outcome? Or?
Yeah, it's identical to that previous trial where you'll be looking at proportion of disease-free patients after the 1 year, trying to get just larger numbers of patients in a trial similar to that one that was done in Taiwan, that trial actually. And it was something like 70 patients in each arm of the trial, so it was quite a substantial phase 2 trial, almost phase 3 anyway.
Well, if there are no more questions, please join me in thanking one more time Professor.